This is my demo for the glucose transport system. On the left, I have the gut lumen. The gut lumen is connected to the epithelial cell by the SGLT1 protein, and the epithelial cell is connected to the extracellular fluid by the sodium-potassium pump. The gut lumen has a large solute concentration, which makes it hypertonic, while the epithelial cell has a smaller solute concentration, which makes it hypotonic. Also, the gut lumen and the extracellular fluid both have a high concentration of sodium. The SGLT1 is a cotransporter that moves sodium down its concentration gradient, which is from high concentration to low concentration. The sodium carries a glucose along with each sodium down its concentration gradient. This is a form of facilitated transport. This process repeats because the concentration of sodium is still high in the gut lumen, and there are still glucose molecules that can be co-transported. Now, the epithelial cell has a high concentration of solutes from the sodium and glucose molecules that were co-transported in. For the SGLT1 cotransporter to continue its cycle, the epithelial cell must maintain a low concentration of sodium. So the sodium-potassium pump helps to regulate that. The sodium-potassium pump is a main mechanism used by cells in order to maintain the concentration gradient of the potassium and sodium ions. Because this process uses ATP, it is an example of active transport. The sodium-potassium first binds 3 sodium ions from the epithelial cell, followed by ATP. The ATP hydrolysis phosphorylates the pump and induces a conformational change. The phosphorylated form has a decreased affinity for sodium, which allows it to diffuse away into the extracellular fluid. The pump then binds 2 potassium ions from the extracellular fluid, which releases the phosphate and induces another conformational change. This then releases potassium and allows the cycle to start again.